Hey everybody, it's your pal Ellen, back with another exciting topic. Today we're diving headfirst into something absolutely huge, a field that's rapidly changing the landscape of science and medicine. We're talking about gene editing. Sounds like sci-fi, right? Something straight out of a futuristic movie? But believe it or not, it's very real. And it's not just real, it's changing our world at an unbelievably fast pace. We're witnessing advancements that were once considered impossible. This isn't just about creating novelty items like glow-in-the-dark hamsters, although that's a fun thought. We're talking about potentially curing diseases that have relentlessly haunted families, passing down through generations, causing immense suffering. Imagine a world with significantly less suffering, a world where these inherited conditions are no longer a life sentence. Think about the impact on diseases like cystic fibrosis or the devastating effects of Huntington's disease. And the applications extend far beyond just humans. Gene editing has the potential to help us grow more resilient and nutritious crops, allowing us to feed a growing global population more effectively and develop sustainable strategies to fight off destructive pests. It's as if we've suddenly unlocked a brand new, incredibly powerful toolkit for manipulating life itself. But as the saying goes, with great power comes an equally great responsibility. We need to proceed with caution and careful consideration. This technology raises a lot of tough questions, ethical dilemmas that we as a society need to grapple with. What are the long-term implications of changing our DNA? And perhaps more importantly, who gets to decide what is acceptable to change and what isn't? This technology is advancing at lightning speed, outpacing our ability to fully understand its potential consequences. Scientists are achieving groundbreaking breakthroughs all the time, pushing the boundaries of what we thought was possible. It's incredibly exciting, but it's crucial that we all take the time to understand exactly what's happening in this rapidly evolving field. Because gene editing isn't just a topic for scientists tucked away in their labs. It's a technology that has the potential to impact each and every one of us, every single one of us. So let's try to figure this out together, explore the possibilities and address the challenges. We're standing at the very beginning of a genetic revolution, a turning point in human history. The choices we make right now, in these early stages, will profoundly shape the future for generations to come. So the question remains, are we truly ready for this immense power? Let's embark on this journey together and find out. Stick with me, we've got a lot to unpack here. We're about to dive deep and explore the fascinating and sometimes unsettling world of gene editing. It's wild, it's undeniably real, and it's all happening right now in this very moment. Ready to jump in? Let's go. This is the future, folks, and it's unfolding before our very eyes. All right, let's break down gene editing. What exactly is it and why is everyone talking about it? Think of your DNA, that iconic double helix, as a giant instruction manual, a comprehensive blueprint for your body. It contains all the instructions needed to build and operate you. Sometimes there's a typo, an error in the code, a tiny mistake, a single misplaced letter that can cause big problems like genetic diseases. These diseases can range in severity, significantly impacting a person's life. Gene editing is like a super precise way to find and fix that typo. It's about correcting those errors at their source. Enter CRISPR, say it with me, CRISPR. This is the star of the show. CRISPR is like a pair of molecular scissors, but way smarter. These aren't your average craft scissors, they're incredibly precise. Scientists can program CRISPR to find a specific spot in your DNA. It's all about targeting the exact location of the error. It's like having a GPS for your genes, guiding you directly to the problem area. Once it finds the target, CRISPR snips the DNA at just the right place. A clean, precise cut. Before CRISPR, editing DNA was clumsy, a bit like trying to fix a watch with a hammer. Now it's surgical, precise, controlled, and effective. After the cut, the cell's natural repair system takes over. The cell recognizes the break and initiates its repair mechanisms. Scientists can help it fix the break, remove a faulty gene, ensuring the error is completely eliminated, or even paste in a new one, replacing the incorrect sequence with a healthy one. So, in a nutshell, let's recap the key components. DNA is your instruction book, the complete guide to building you. CRISPR is your editing tool, the molecular scissors that can make precise changes. And now we can make precise changes, correcting errors and potentially curing diseases. It's powerful, it's precise, a revolutionary tool in the world of genetics. And it's opening up a universe of possibilities, from treating diseases to enhancing human capabilities. But with this power, we have to be careful. Ethical considerations are paramount. How did we get here? It's a wild story filled with twists and turns. Let's rewind and see how gene editing went from pea plants, the foundation of genetics, to Nobel Prizes, the ultimate recognition of scientific achievement. Ready for a quick history lesson? It's more exciting than you think. Let's jump in. The water's fine. The journey is just as fascinating as the science. It's a story of human ingenuity and perseverance. Stay with me. You won't want to miss this. Here we go. Let's dive into the past and uncover the origins of gene editing. Gene editing didn't just appear overnight. It started in the 1800s with Gregor Mendel and his pea plants. He discovered the basics of inheritance. Fast forward to the 1950s, Watson, Crick, Franklin, and Wilkins revealed DNA's double helix structure, the blueprint of life. Suddenly, we could see what genes were made of. Then came the Human Genome Project, mapping all our genes, 3 billion letters. This gave us a reference book for human genetics and helped us link genes to diseases. And then, the CRISPR breakthrough. In 2020, Emmanuelle Charpentier and Jennifer Doudna won the Nobel Prize for developing CRISPR-Cas9. They turned a natural bacterial system into precise genetic scissors. Gene editing became faster, cheaper, and more accessible. From peas to Nobel Prizes, it's been a wild ride. Now, let's see how we're using this power today. The future is here. Let's check it out. Gene editing is no longer a futuristic fantasy. It's actively changing lives in the present day. We're seeing its impact across various fields, offering solutions and raising important questions. In medicine, CRISPR is rapidly becoming a game changer, being used to tackle some of the toughest genetic diseases that have plagued humanity for generations. Diseases like sickle cell anemia, cystic fibrosis, which cause immense suffering. And the fight against cancer is also seeing new hope with gene editing, even some cancers. Early trials are showing real promise. We're seeing encouraging results that suggest a brighter future for patients. Maybe even cures, not just treatments that manage symptoms, but actual cures that eradicate the disease at its source. In agriculture, gene editing is revolutionizing the way we grow food, making crops more resilient to pests, diseases, and climate change. And more nutritious, packed with essential vitamins and minerals. Imagine rice with more vitamins, a staple food fortified to combat malnutrition in vulnerable populations. Or tomatoes that taste better, more flavorful and appealing to consumers. This could lead to feeding more people, fighting hunger on a global scale, and ensuring food security for future generations. But then came the whoa moment, a stark reminder of the ethical considerations surrounding this powerful technology. In 2018, a scientist in China, acting outside the bounds of established ethical norms, announced the first gene-edited babies, a move that sent shockwaves through the scientific community and beyond. He tried to make them resistant to HIV, a noble goal perhaps, but did it secretly and crossed ethical lines, raising serious questions about the responsible use of gene editing. The world reacted with shock and concern, highlighting the need for global guidelines and oversight. Editing embryos means changes can be passed down forever, altering the human germline. That's a huge step and a huge risk, with potentially unforeseen consequences for future generations. Now, there's more caution and debate, a renewed focus on ethical considerations and responsible innovation about where to draw the line, what boundaries should be in place to prevent misuse and ensure safety, what's okay to edit, what's not. These are the fundamental questions we must grapple with, especially when it comes to changes that can be inherited, impacting not just individuals but entire family lineages. The potential is enormous, offering solutions to some of humanity's greatest challenges, but so are the responsibilities, demanding careful consideration and ethical oversight. We're walking a tricky path, navigating uncharted territory with both excitement and trepidation. The conversation is just getting started, and it's a conversation that needs to involve everyone. Let's talk about the big questions we all need to ask, the ethical dilemmas and societal implications that demand our attention. Ready? Let's dive in. Gene editing's promise is huge, but so are the ethical dilemmas. Editing human embryos, germline editing, means permanent changes to the human gene pool. That's a heavy responsibility. Where do we draw the line between therapy and enhancement? Curing a disease is one thing, but making someone taller or smarter? That's a slippery slope. The idea of designer babies is no longer just sci-fi. Could gene editing create new social inequalities? Genetically enhanced versus unenhanced? Access and fairness are big concerns. We don't want a genetic class system. There are risks too. What if fixing one gene causes new problems? Unintended consequences could last for generations. We're dealing with the building blocks of life. We need humility about what we don't know. These are conversations we all need to have, openly and honestly. The decisions we make now will shape humanity's future. No pressure, right? But it's true, let's keep asking the tough questions. Our future depends on it. Gene editing won't just stay in the lab, it'll touch every part of our lives. Education will need to keep up: more scientists, more genetic counselors, more ethics classes. Healthcare could be revolutionized, personalized medicine treatments tailored to your DNA. But will these therapies be affordable for everyone or just the wealthy? New industries and jobs will emerge, but others may disappear. We'll all need to learn a bit about genetics no matter our job. Governments will have to create smart laws and regulations. How do we ensure safety, privacy, and fairness? International competition will heat up: what one country allows, another might ban. Parents will face new choices about their children's health and traits. Society is about to change in ways we can barely imagine. The ripple effects will be everywhere. We need to be ready. The future is coming fast. Let's make sure it's a future for everyone. We all have a role to play. Let's step up. Gene editing isn't just about bodies, it's about identity. If you were edited to be good at math or avoid a disease, does that change how you see yourself? What happens to our uniqueness if we start fixing imperfections? Could we lose the diversity that makes us interesting? New anxieties could emerge: pressure to edit, fear of being left behind. What about those born naturally? Will they feel different or disadvantaged? Gene editing challenges what it means to be human. What is normal? What is natural? Are we just genes to be optimized, or is there more to us? These are questions we'll be asking for a long time. It's not just science, it's about our soul. Let's keep the conversation going. We have a powerful tool: gene editing. Now we need wise regulation, not too strict to block progress, but strong enough to keep us safe and ethical. It's a global issue. Science crosses borders, so our rules should too. We need international cooperation and shared standards. No country should become a haven for risky experiments. Teamwork is essential. Public understanding matters just as much. This isn't just for scientists or politicians. It affects all of us. We all need to be part of the conversation. Learn, talk, and make your voice heard. The more we understand, the better our decisions will be. It's about balancing hope with caution. We need to encourage research that helps people, but set guardrails to prevent harm. It's a tough balancing act, but we can do it. The future depends on thoughtful choices. Let's move forward carefully, together. Knowledge is power. Let's use it wisely. We're all in this. The next chapter is up to us. Let's write it well. We can do this. We're at a turning point in human history. Gene editing gives us the power to rewrite life itself. The potential is incredible: curing diseases, feeding the world, understanding life in new ways, but the risks are real. Designer babies, new inequalities, and questions about what it means to be human. The future of gene editing isn't set in stone. It will be shaped by the choices we make now. Scientists, doctors, policymakers, and everyday people all have a role. We need to be informed, engaged, and demand transparency. These tools must benefit everyone, not just a few. It's a big responsibility and an amazing opportunity. As we stand on the brink of this new era, we have to ask, what kind of future do we want to create? What world will we leave for our children? The future is in our hands and in our genes. Let's choose wisely. Let's build a future we can be proud of. The story isn't finished. It starts with us.